Yeah.
Okay, very good evening, my dear friends. Regarding of this CPGET 2023 free online class, today's our eminent resource person, Dr. Deshaboyina Nagaraju garu, eminent mycologist, assistant professor, Department of Botany, City College, Government City College Autonomous, Nayapur, Hyderabad. Sir will going to explain about the important area of the mycology, especially the general characteristic features of the fungi. So in depth and in detailed, and all the points covered during this period, during this time. So my dear friends, better you have to utilize this wonderful opportunity. It will help you crash the CPGET 2023 and also other competitive exams like say JL, DL and Gurukul PGT, JL, DL also. So my dear friends, so this is a wonderful opportunity. So we would like to invite our today's eminent resource person, Dr. D. Nagaraju. Please welcome, sir, and start your session, sir. Please welcome.
Okay. Good evening to all. Is it audible?
Yes, sir.
Good evening to all.
Respond.
Good evening, sir.
Respond to this, sir. All of you respond, my dear friends.
Good morning. Good evening, sir.
As we are discussing the fungi, classes. Now, regarding the type studies, you know, the most of the fungi are saprophytes. I already told you. Only few are parasites. As a saprophyte, these are helping to clean the nature and to add the minerals to the soil. Okay, but very few are parasites. You know, we have studied so many. What are those for each group? For the zygomycotina, for the mastomycotina, zygomycotina, ascomycotina, basidiomycotina, and deuteromycotina. For each one, at least one or two, you have studied in the degree level, UG level. That all are somewhat specifications are there for that. You know, if you take the mastomycotina, what we have studied in the mastomycotina? Do you have any idea?
Phytophthora.
Phytophthora, Pythium, Phytophthora, yeah, and Albugo you are not studied, huh?
Albugo candida, sir. Albugos.
Albugo or system.
Stop. Yes.
Albugo it causes what disease it is? White rust. White rust, white patches upon the leaves of especially Brassicaceae, Orthocaceae, Chenopodiaceae family members. You know, there is question raised, white rust of whatever it is, Cruciferia or Podostomaceae, Chenopodiaceae, family, especially upon the leaves. White spot. That caused by? Caused by? Albugo. What it is? Huh? Albugo? Yeah. Albugo candida or albugo blüti. Based upon the host, so many specific diseases there. The splitted leaf, white, only white splitted spots, or pustules, various spots are there. But you have to remember only thing is here the by albugo. Okay. Albugo candidiasis, it causes the disease white rust. Okay. But white rust disease caused by
Brassica.
Albuco candida.
Albugo candida.
which plant?
Brassica family.
That is
Brassica nigra.
Rakam capital Brassica. Diagram. Okay. Brassica, it have what type of the life cycle? Brassica life cycle is what type it is? You see here what it is, haplobiontic. That means it having applied and diploid, both the phases are there. Usually it is the lower fungi. That's why the sexual reproduction, plasmogamy, karyogamy, meiosis both takes place immediately one after other. There is no gap. Here, the master fungi are called as zoosporic fungi, which release the spores with the flagella. Yes or not? You see here, whatever the pustules, that again releasing the juice spores, the juice spore germinate and it affect on the infects upon the other leaf, so that it widespreads upon the field wherever it is. Okay. And then in the next zygomystis life cycle, you go for the zygomystis life cycle. Zygomystis are cenocytic. These are cenocytic thallus. Most of the zygomystis are cenocytic thallus. That means it having the numerous cells. Numerous. What are there? Numerous cells or nucleus? Cells or nucleus? Nuclei. Numerous nuclei are present. That means more number of the nucleus are present without the septa. That's why cenocytic. I already told you. Huh? These cenocytic thallus. You know here one more thing. Sporangiospore and zygospore. There is a difference. Sporangiospore and zygospore. See the what is the difference between sporangiospore and zygospore? You have any idea? Sporangiospore and zygospore. Sporangiospore. Sporangiospores, which are formed in the asexual reproduction directly in the sporangia. You know the mycelial structure. Mycelial structure of the Rhizopus mucor. What are those?
Cenocytic.
Sporangiospore are there now. Rhizoid, mycelium, sporangiospore. These upon the sporangiospore, spores are released. That called as sporangiospores, which are there in the sporangia. That's why sporangiospores. And then in sexual reproduction, what type of the sexual reproduction it is? Gametangial, contact, gametangial copulation. Gametangial copulation. What type it is?
Gametangial copulation.
Copulation. Gametangial contact is there in the masticomycotina. gametangial contact type of the sexual reproduction in the Masticomycotina. There you might have seen. You have to cut it, huh? Here you can see the two. That means, whatever it is, the male and female, oogonium and antheridium, both are side by side. Whereas here, the opposite stain, plus and minus 10, opposite stain, thallus, it's from the pro-gametangia. The two comes together and it melt the whatever the gametangial wall and it from the zygo, whatever it is, what it is.
Zygos.
You see this here, you take the reduction division and it form haploid spores. You know, columella is present in columella. Where the columella is present? In the legs, huh? Columella. Where? In the sporangiospore. Here you can see the hyaline space of the sporangiophore, where the whole nucleus and cytoplasm will go to the top, and the remaining part, the separating part, it's separate with the collar.
Capsule. In
Capsule.
Mm.
What's the capsule? The capsule is there.
Food zeta in capsule is the
Collar shape structure is there. This collar is divide the fertile part from the vegetative part. The collar, that collar is called as collar. The collar, the separating layer, as like a set collar at the neck. Here you can see the sporangias which are fully loaded with the sporangia nucleus. That above is the what it is? Sporangiospores. Okay. And with the gametangial copulation, we take the reduction, sexual reproduction, and it forms the zygospores. The zygospores germinate, and again it forms the sporangium and sporangiospores. In the sporangium, the sporangium germinate, and it forms the new thallus. Okay. Now here in the zygomycotina, actually these are totally saprohytes. Very few are weak parasites. You know the mucor, rhizopus, you know the bread mold, black mold, pin mold. Majority of them are saprohytes. But in the recent time, mucor that affected COVID patients, those who having the diabetes, sugar, the mucor, especially this mucor rhizopus, which grown in the carbohydrate-rich sources, where the carbohydrate-rich source, that means where the glucose level is high in the blood, that candidate are affected with this mucormycosis, skin infections, eyes, whatever the sensitive parts and the blood vessels, where the delicate blood vessels are there, it damages the tissues. This black mucormycosis. With the mucormycosis also, so many are died. So many COVID patients are died, even after curing from the COVID. Okay. And next, ascomycotina. Ascomycetes life cycle. What are those? Ascomycetes, especially, you know, I already told you, in the crow's ear, the curved hairpin shape structure, nuclear fusion takes place. That means karyogamy will take place in the crow's ears. I already told you. After that, ascospores are formed. The ascus, in each ascus, how many ascospores are there? 8 ascospores are formed. That all you should remember. Crozier, in each ascus, 8 ascospores. And what are the fructification? That means fruiting bodies of the ascomystis. That means ascocarp. Carp means fruiting body of ascomystis. That's why ascocarp. You know, I already discussed the clistothecium, the round, unopened fruiting body, and you know the perithecium, the flask-shaped fruiting body, apothecium, saucer-shaped fruiting body, pseudothecium or ascostroma, perithecia-like, but there is no clear peridium. Okay, these are the 4 type of the fruiting bodies are there in the ascomycotina. What happened? And then, you know, very, very important, the Puccinia life cycle in the Basidiomystis. You know, in the ascomystis, what are there? Yeast, Saccharomyces cerevisiae, yeast. You know, the budding, by budding, the yeast will take the asexual reproduction by budding. The majorly it form it proliferate by the budding. That means it increase their number by the budding, and also by the conjugation mechanism. That means two yeasts that are comes together and joined it, and it form a zygote, like form the applied spores. That applied spores will germinate and it form the new yeasts. Okay. And then one more is in the ascomycotina is penicillium. We have studied now. You know, Alexander Fleming. That most important question that we will discuss in the academic important. Alexander Fleming discovered, separated, identified the penicillin from the Penicillium notatum. You know the Penicillium notatum and Penicillium chrysogenum. These are the very prominent Penicillium species. You know the Penicillium, it is a biverticillate. That means it having the triangula, metale, and conidiophores. That means on the second branch, that means on the medullae, the conidiophores are formed. That's why it look like a brush. And you know, penicillium also it forming the clistothecium, unopened, round, ball-shaped fruiting body. That means penicillium fruiting body is clistothecium. Clistothecium. You know the perfect stage of the penicillium. Perfect stage of penicillium. Already told you. Perfect stage of aspergillus and perfect stage of penicillium. These two I already told you. Toleromyces. The perfect stage of penicillium is toleromyces. Okay. And you know the next is basidiomyces. In the basidiomyces, you know basidiospores, clamp connections, dolichosepta. These are the very, very important. Dolichosepta, peranthozomes. Do peranthozomes is attached at the septa and it having the open porous space with the semi-permeability. That is a special character. And it having the primary, secondary, and tertiary mycelium. Tertiary mycelium is nothing but basidium, fruiting body. Tertiary mycelium is nothing but the basidium, the fruiting body, button-shaped fruiting bodies. You know the button-shaped mushrooms, toadstools, puffballs, bracket fungi. geosters, so many visible fruiting bodies which are available on the soil. Most of them are basidiomycetes, very few are ascomycetes. Okay, and then here one more thing also I told you, clamp connections. Clamp connection, bypass, outer ring roads. That means the outer extra projections like pin are like clamp, are like clip structures which interconnect between the two cells. That bypass the interconnecting one from the interconnecting one nucleus from the one cell to other cell will go and mate so that dichordization will take place. You know the dichordization, that means two nucleus in each cell. Dichordization is take place because of clamp connections. Primary mycelium, you know the basidiomycetes fungi. Is there any sexual organs are there? The male and female oogonium and antheridium like structures? No. Here the vegetative thallus only will mate. That type of the reproduction what we call somatogamy. Somatogamy, you might have forgotten. Somatogamy means somatic. That means vegetative. Vegetative thallus are fusing and it forming the fruiting body. Primary mycelium by mating it form the secondary mycelium. The secondary mycelium by forming the clamp connections it form the karyogamy. So that it form the tertiary mycelium, that nothing but fruiting body, that we also call it as basidium. You know the basidium two types also I discussed, na? Hollow basidium and progmobasidium. Hollow basidium means the basidium without the septa. Basidium without the septa, basidium it having the ball shape sac like structures that are hollow basidium. Usually most of them are hollow basidium, but very few are progmobasidium. You see the progmobasidium in the Puccinia. That means it having the branches. Basidium it are dividing into branches. That branch basidium is called as progmobasidium. Progmobasidium, which is the interface in between the two hosts, primary host and secondary host. The basidiospores are formed in the Puccinia. These are developed upon the soil and again it attacks on the secondary host or alternate host. Progmobasidium also called as what? Teleomycetes. That means teleobasidium. We also call it teleobasidium or progmobasidium. Okay. And how many basidiospores are formed from basidium?
4, sir.
4. Even in the progmobasidium or hollow basidium, 4 basidiospores are formed. The ascus 8 ascospore. The basidium 4 basidiospores. Okay. Basidiospores, whenever it releasing basidiospores, it having the buller mechanism. You know the buller mechanism? There the basidiospore, there it the water drops. It attached with the water drop. Whenever the water drop is added at this basidiospore space, with the water drop, the basidiospore will be detached from the basidiophore. That we call it as what it is?
Fuller.
Buller mechanism. Okay. That most important, buller mechanism. And you know the Puccinia life cycle. also called as macrocyclic life cycle. That means we're having a very lengthy, large life cycle. That's why we call it as macrocyclic. They're having the 5 type of the spores in the life cycle. You know, it depend on 2 hosts. That means 2 houses. That's why also called as heteroecious. Heteroecious, 2 hosts, primary host and secondary host. The primary host where the reduction division, spores formation, that means sexual cycle like structure will take place. That is the primary host. Where the asexual spores are forming, that are called as secondary host or alternate host. Okay. Which spores attack on the wheat? Puccinia, what type of the spores? Whatever the Puccinia spores attack on the wheat. ECS spores, sir. Igneous spores, sir. Whatever the spores. Which spores it attack on wheat? What type of the spores are present in the primary host? What type of the spores are present in the secondary host or alternate host? This question raised so many times. What type of the spores?
Basidiospores.
Basidiospores. Basidiospores are not there in any host. Huh? Actually, what is the fungi which cause the wheat rust? Puccinia graminis triticide. Puccinia
Gramin.
triticide fungi which cause
Wheat rust.
wheat rust. Wheat rust caused by Puccinia graminis triticide. Not only on the wheat, other hosts also will be affected with the Puccinia. Puccinia recondita, Puccinia stripfly, so many are there. Okay. Here in the Puccinia graminis triticide, which cause the wheat rust. On the primary host, what are the spores are present on the primary host? And which spore attack on the wheat? This is most important. You already studied this all now. Which spore which attack? Huh? Which spore? You don't know? You have to interact.
Ichthyospores.
Ichthyospores. Whenever it having the primary host, that means wheat, that attack the wheat and it form the and it generate the what type of the spores? What type of the spores? Uridospores. Red color. Urido means red. Red patches upon the leaf. Red patches upon the leaf. That because of these mycelial spores which are in red in color, that's why called as. That's why we called it as. Called it as nothing, huh? Uridospores, red color spores. That's why we call it as uridospores. Here you can see these are binucleate. You see the 2 nucleus are present. Binucleate spores. That means dikaryotic mycelium. These spores again it cause the disease to the whole wheat crop or to the next plant nearby or surrounding plant. That's why we also call it as repeating spores. Iridospores also called as
Eating spores.
repeating spores. That means it repeats the disease. It causes the wide spreading of the disease. It causes the wide spreading of the disease. That's why we call it as repeating spores. Repeating spores. What are the repeating spores in the Puccinia? Iridospores, Iridospores, Basidiospores. You know the on the Puccinia, we trust K.C. Mehta has studied. Who studied elaborately on the wheat rust disease? Who
Yes.
invented, that means who identified the disease causing mechanism? Is K.C. Mehta. K.C. Mehta study you also studied, na?
Yes, sir.
Okay, next, iridospores. It repeats the disease, and also whenever it gets mature, it becomes turned into black color. That's why we call it as heliospore. Helio means black. Irido means red. Heliospores. Here, whatever the nucleus, two nucleus, that means two dikaryotic phase is there. The dikaryotic phase where by forming the septa, it are separated. That means two cells. Heliospores are two cell spores. Okay. These heliospores, which again not attack on the wheat. You know, iridospores also called as resting spores. Some spores, resting spores, repeating spores. But teleospores which will not attack again on the wheat. The teleospores which take germination by forming the basidiospores. By forming the basidiospores, these basidiospores are spread in the air. You know the what type of the basidiospores it is? Pragmobasidium, teleobasidium. Pragmobasidium or teleobasidium. Why it having the fragments? That means branch. Pragmobasidium, branched basidium. That's why we call it pragmobasidium. The pragmobasidium which release the basidiospores, these basidiospores also will not attack on the primary host. The secondary host, alternate host, weed plant. What happened? What happened? What you are doing? Okay, these basidiospores will attack on the alternate host. Which is the alternate host of the Puccinia in the wheat rust? What is the secondary host? What is the alternate host? Barbary plant. I already told you Barbary plant. The alternate host or secondary host, not the Barbary plant. The Barbary plant, it is a wheat plant. If it is grown nearby surroundings of the wheat, it carries the spores. The spores will get mature and it forms the cause for the formation of the esiospores. That again esiospores will attack on the primary host. You know that whenever the basidiospores, if it fall upon the lower side, upper side of the, that means adaxial surface of the leaf, it forms the flask shape fruiting body structure. That what we call it as flask shape fruiting body is called as Huh? Just now I told you now, flask shape rooting body, saucer shape rooting body.
Apothecium.
Apothecium is saucer shape. Perithecium. Right, from the spores, plenty of the spores. The spores are like a spermaceospores. That's why we call it as spermaceum. These spermace here, it also having the receptive hyphae as like a female organ. The receptive hyphae which mate with the spermaceum and it from the next spores that we call it as esiospores are also called as peridiospores are also called as pycnidiospores. Esiospores or pycnidiospores. These at the same time on the lower side esiospores or pycnidiospores are formed, on the upper side the spermaceums are formed. These ECMs, one more important thing is that how the esiospores are there, how the ECM is a reverse, that means the bell shape, reverse U shape or bell shape structure. There the esiospores or pycnidia are like a chain. This ECM will not attack on the secondary host, Barbary plant, will not affect on the Barbary plant. If the primary host, wheat is there, again it attack on the wheat crop. Okay. So that the life cycle will be completed. If we remove the secondary host, the spore formation will be stopped at the up to only the teleospores only, basidiospores only. So that we can control by removing the weeds. And also, you know, the Casey Mehta said with the monsoons, from the northeast monsoons on the southwest monsoons, the spores are coming from north to south and south to north. By stopping the cultivation of the any area, either in the south or in the north, up to 1 to 2 years, they could able to stop the widespreading of this disease. The very simple ecological balance system by environmental system, by following the environmental systems, simple systems, they could able to control the wheat rust disease. That special method, the special causes identified by the Casey Mehta and his associations, Agra University, Casey Mehta and his associations. The main problem identified, so that mostly in our southern area, we are not cultivating the wheat. All in the north, if the whole India cultivate the spores, iridospores, whenever it comes the iridospores in the September and October time, that again come to here, where here in the crop are in the fertile conditions, then here it attack, and again in the southwest monsoon, these whatever the spores, iridospores which are there in the south area, that will go and attack on the north side, so that it disease will be repeated. We can't control. So there is an alternate method of the cultivation that is noted by Casey Mehta. Okay, here in the Puccinia, most important is Puccinia life cycle, also called as macrocyclic or heteroecious life cycle. Okay, what type of the spores which attack on the wheat? Iridospores, teledospores, and esiospores. Okay, what type of the spores are formed on the secondary host or alternate host? What is the secondary host? What is the alternate host? Barbary. What are the spores which are forming on the alternate host? Spermaceum and ecium. Spermaceospores and esiospores, also called as pycnidiospores. Okay. And what are the diploid spores? What are the haploid spores? What are the diploid spores? Iridospore, tildospores are diploid. In the basidium, prochnobasidium, here is the reduction division which takes place. So from here, the haploid spores, basidiospores, ichthyospores, pycnospores, basidiospores, spermaciospores. Up to this, these two are haploid. Again, ichthyospores here, petalization is taken place with the receptive hyphae and spermacia. Again, the diploid spores are formed in the pycnidium, ecium. So, ecium, basidiospores, tildospores, ichthyospores, basidiospore, tildospore. These three are diploid, and basidiospores, spermaciospores are haploid. This is also very important. Diploid spores, haploid spores. Which among is not the diploid spore? Ichthyospore, basidiospore, teleospore, iridospores. Which one? Which among is not the diploid spore? Ichthyospores, iridospore, tildospore, basidiospores. Which among?
Basidiospores, sir.
Basidiospores. Remaining three are diploid. Basidiospores, why? Here in the Pragmobasidium, basidium is taken the reduction division. That's why that are haploid. Okay. Oh yeah, again live. You have to go for the economic importance, no? Okay. Then where we have Saccharomyces life cycle is also already gone, completed, no? What happened? Okay. The penicillium life cycle also we already told you. Here the medullae, phyllodes, conidium, frangula, medullae. That's already told you. Here the gametangel copulation. Gametangel, you see the clistothecium. What are the fruiting body in the penicillium? Clistothecium, round circular. There is no force. And what is other? This one. Cercospora also. Deuteromycotina, imperfect fungi, dustbin fungi. We have studied the Cercospora na? In the Deuteromystes. Dustbin fungi. Deuteromystes fungi. Dustbin fungi. Okay. Imperfect fungi. There is no perfect stage. If we identify the perfect stage based upon the fruiting body, we can reallocate into either Masticomycotina, Zygomycotina, Ascomycotina, Basidiomycotina based upon the spores types. You know the thick disease of groundnut, leaf spot of groundnut caused by Cercospora. No, two type of the leaf spot disease are there. Round circular patches and uneven shape patches of the leaf spots. Cercospora personata, Cercospora arachidicola. Round circular patches are caused by Cercospora personata. Uneven shape patches caused by Cercospora arachidicola. You know the perfect stage of Cercospora. Your question also raised. The perfect stage is Mycosporella. Mycosporella is the perfect stage of Cercospora. That's why the ticka disease of groundnut caused by Mycosporella. Mycosporella. That means Cercospora personata renamed as Mycosporella brachelli. Brachelli is a scientist. And the Cercospora arachidicola renamed as Mycosporella arachidicola. Okay. This is called the leaf spot disease. The Hastoria which are enter with the Hastorias which enter inside the host through the stomata and widespread in the fruiting body inside, that means just below the epidermis, And there it formed the what it is, the fruiting body. Here in the fruiting body, what are there? The septate conidias are present. Conidia which having the septa. Conidia which having the septa. Septate conidia. You know then the coltotrichum, sickle shape. Sickle shape conidia are present. Coltotrichum. You know the coltotrichum which cause the disease?
Sugarcane.
Sugarcane. Red rot of sugarcane caused by coltotrichum palcatum. Sickle shape. Conidiospores are sickle shape. Okay. These all we already discussed now. Where the zygomycetes life cycle. Here where the plasmogamy, where the karyogamy, where the meiosis is taking place. Here in all the lower group of the fungi, plasmogamy, karyogamy, meiosis will take place immediately one after another. There is no gap space. But in the higher fungi, Ascomycina, Bacillomycina, there is a gap between the plasmogamy, karyogamy, and meiosis. These three steps commonly called as sexual reproduction: plasmogamy, karyogamy, meiosis. Whereas in the Ascomycina, you see, after plasmogamy, dichordation, disporic fungi are forming. There in the Ascoscrogius, the karyogamy is taking place. You see, and then the meiosis. Then again it forming the haploid spores. How many? 8 spores. That spores will germinate and it forming the new thallus. You see there is a much gap in between the plasmogamy and karyogamy. Karyogamy will take place in the Crogius like fruiting bodies. Okay. And in the Basidiomycina, there is no sexual organs, only vegetative mycelium. Somatogamy. You see the primary myceliums are mating and it forming the secondary mycelium. In the secondary mycelium, there is a clamp connections which is forming, so that there is a karyogamy is taken place. And then again in the meiosis in the basidium, that means in the tertiary mycelium, floating body in the basidium. Then this release the haploid spores. The haploid spores again germinate and it form the new mycelium. Okay. Up to this is the general characters of the fungi. Okay. Then shall we go for the economic importance or tomorrow? Here are the life cycle, very, very important life cycles. Aplontic, aplodiplantic, and diplobiontic. And the diseases also white rust, wheat rust, Tika disease. These 3 diseases are you have studied. Okay. And then shall we go for the economic importance? Or tomorrow we will. Tomorrow we will discuss the economic importance of the fungi. Okay.
Okay, sir.
Okay. So much are there. Very, very useful. Fungi are economic, agriculture, environmental related, food, industry, medicinal, so many point of view, so much of the agriculture, so much of the importance is there for the fungi that we will discuss tomorrow. Okay.
Right, sir. Really wonderful explanation, sir. Each and every point covered, all the spores, all the life cycles of the different classes of the fungi, from Phycomycetes to Myxomycetes or the Deuteromycotina also. So, my dear friends, regarding of this class, if you have any doubts, please ask our respected resource person, Dr. D. Nagaraju garu. ఏదైనా డౌట్ ఉంటే సార్ని అడగండి.
Any doubts? Any clarifications are needed? How the question raised, you have to focus on it. Tomorrow we will discuss the economic importance of the fungi. Okay, any doubt, any clarifications?
మాట్లాడండి.
Here we have discussed life cycles, type studies only today.
Yes.
Life cycle, type studies of the fungi. Tomorrow we will discuss the economic importance of the fungi. If you have any doubt on last classes, also you can ask. Manoj sir is there, huh?
Good evening, sir.
Good evening, sir. How are you?
Fine, sir. Come on, sir.
మనోజ్ సార్ ఎక్కడి నుంచి సార్?
Come on, sir.
ఖమ్మం.
Yes, sir. Yes, sir.
ఓకే ఓకే.
Any
బాగున్నా సార్.
clarifications, students? What is the life cycle? Huh? Which type of the spores attack? What are the diseases? What are the primary hosts? What disease caused by which fungi? Huh? Yeast or penicillium belongs to Ascomycotina, Basidiomycotina, Deuteromycotina. Yeast and penicillium belongs to yeast and penicillium. This group, which group? Ascomycotina.
Ascomycotina.
Ascomycotina. Which one? Ascomycotina. Ascus. Ascomycetes. And the Puccinia belongs to which group?
Basidio.
Yeah.
Basidiomycetes. Basidiomycotina. Basidiom bracket fungi, puffballs, turtles. Huh? What are the other names are there for the Basidiomycotina? Toadstools are Basidiomycetes, Ascomycetes,
Packet. And bread mold.
Basidiomycetes. That means what I ask you, toadstools, puffballs, or polypores, earth stars, birds, fungi, or rust, or smuts. These all are belongs to Basidiomycetes fungi. Rust fungi belongs to which group? Rust fungi.
Uridinales.
Uridinales order group is Basidiomycetes. And the smut fungi belongs to Eustyloginales. Smut caused by Uridinales, Eustyloginales, Peronosporales, like that they ask the question.
Peronosporales down mildew diseases.
Yeah, downy mildew disease caused by
Peronosporels.
peronosporels.
Okay, sir. Thank you.
Smut disease is caused by eustylogenales. Rust disease caused by uridinales. That question already raised so many times. Rust disease caused by uridinales, eustylogenales, peronosporels, mycosporels, whatever it is. Smut disease is caused by, or smut belongs to
Yes.
mycotina. Okay. And you know about the yeast and penicillium. These two highly valuable for the industry and medicinal purposes. You know, without the yeast, there is no bakery, no beverages. Okay. Any doubts?
Respond, my dear friends. Any doubts?
You know the Copropillus fungi. Copropillus fungi. Which belongs
Dung, sir. Cow dung or
The fungus which grown on cow dung or cattle.
dung.
that belongs to Ascomycetes. Copropillus fungi. That question also raising so many times. And any which among is not the ascocarp? Perithecium, apothecium, clistothecium.
Clistothecium, sir.
These are ascocarps.
Yes. Perithecium.
Clistothecium is the fruiting body of penicillium. I already told you. Huh? Just now I told you clistothecium is the fruiting body of penicillium. The round, circular, without porous.
Yes, sir.
Huh?
Yes, sir.
Just now I told, but you forgot. Clistothecium is not ascocarpa.
Ascocarp.
Clistothecium, apothecium, these three are pseudothecium. These are all are ascocarp, fruiting body of Ascomycetes. Don't forget. Just now in this class only I told. The question raised already. Penicillium fruiting body is Clistothecium. Okay. And any doubt? Shall we stop? Huh?
Yes, sir. Thank you very much, sir, for your
Okay, right.
valuable sparing of your valuable services for our
More class.
young aspirants.
The economic importance, that is very, very important.
Okay, tomorrow same time, sir. Okay, sir. Thank you, sir. Thank you. So your valuable time is very, very valuable services, sir. So young aspirants definitely they are get benefits from your valuable today's class, sir. Thank you very much. Tomorrow same time we will going to meet, sir. Thank you.
Okay then.
Right, my dear friends.